Hello students, now we will start with a new topic, that is semi-conservation of DNA replication or semi-conservation mode of DNA replication. This method was given by Michelson and Stahl. In 1958, while they are working on E. coli. Work on E. coli. You know that E. coli is an anaerobic and gram-negative bacteria. What is aim of this experiment? It is to prove that DNA always tries to conserve or preserve half of its genetic material. In the sense, if there are two strands, each strand tries to protect its own genetic material by giving rise to new DNA. That is meaning of this semi-conservation method of DNA replication. Aim is to prove that at least half of the genetic material, that is DNA strand, tries to protect its own genetic material. Double-stranded DNA tries to preserve its genetic information. For this method, they have invented a cesium chloride or density gradient centrifugation method. So they invented density gradient centrifugation method. What is speciality of this method? In case of centrifugation, when we put the cuvettes or the test tubes in the centrifuge machine, the rotations here, according to this experiment, is 6 lakhs rotations per minute. So that is nothing but the centrifugation. Then what is meant by gradient, density gradient? Here, when some types of particles are kept in some other types of solutions. For example, when DNA is kept in the reagent cesium chloride, then cesium chloride has the nature of allowing DNA particles to get separated basing on the density of the DNA particles, because normally DNA will be having N14 in the nitrogen bases. Nitrogen atoms, in case of purines, it will be 1, 3, 7, 9 positions of the ring, and in case of pyrimidines, it will be 1 and 3rd positions of the rings. But these people have introduced N15, that is heavy nitrogen atom into the purines and pyrimidines, nothing but nitrogen bases. It is a heavy nitrogen but not radioisotope nitrogen. It is a heavy nitrogen but not radioisotope. So when such type of DNA are having this type of nitrogens and this type of nitrogen in same strands or in different strands, then the strands that are containing this heavy nitrogen and this normal nitrogen will be separated based on their densities. That I will give you in the form of a sentence. This method is also called as buoyant density method. What is meant by this? It means a measure of the tendency of a substance, to float in some other mixture. Here, the tendency of the substance I have written, it means the DNA has the nature to float in some other mixture. It means here we can consider cesium chloride. Means DNA is getting floated or separated based on different densities. That density differences are because of nitrogen 15. One more point. In this experiment, here we have written when they are working on E. coli. So E. coli divides by binary fission for every 20 minutes. Messelson and Stahl has also considered very particularly about the timing that is taken by the E. coli for its replication. It means that if one E. coli is getting divided into its offsprings or getting replicated by the process of binary fission, it means that in the DNA which is present inside its body also gets replicated. That concept was also concluded in this experiment. Density gradient centrifugation is a method of separating DNA strands or molecules basing on their density. There is another method, that is differential centrifugation. In that method, DNA particles are separated basing on their size and also the shape. Here, cesium chloride is used as a reagent. Now, cesium chloride has a nature of separating DNA strands based on density only, but not based on size and shape. So, if we write a diagrammatic representation of this, we can write, first we will write one point, Messelson and Stahl has used nitrogen 15 in the medium where E. coli is grown. This N15 is a heavy nitrogen but not radioisotope nitrogen. Now, so when the cesium chloride plus DNA with this type of nitrogen and DNA with normal nitrogen are kept in a test tube and allowed for centrifugation, then N15 containing DNA are at the bottom because of their high density. N14 and N15 means intermediate DNA strands, which have both of these, are present in the middle area above the bottom part, and purely nitrogen 14 containing DNA strands, they are present on the topmost area of the test tube. So here one band, here the other band, here other band. So all these bands are differentiated basing on the density. The density here at the bottom is more. The density of the bands at upper means top of the test tube is less density. The bands are separated based on density. We will see how the timing is taken by the E. coli to divide and how these DNA of different densities got separated and how many number of this intermediate hybrid DNA have been obtained, how many number of N14 containing DNA are obtained. Are there any names given for the DNA strands that are containing only N15, only N14? So, initially, E. coli are grown in medium with N15. So, all strands of DNA have only N15 type of nitrogen called as heavy DNA. Heavy DNA. This we can write like, so this strand of DNA in E. coli has N15. This strand of DNA has N15. Think that these both are present in an E. coli DNA strands. E. coli takes 20 minutes for its replication. So, after 20 minutes, we'll write this one in a neat diagrammatic representation. E. coli with N15 in both the double-stranded DNA. So, it has taken 20 minutes for its division. The DNA with both strands N15 containing nitrogen atoms is called as heavy DNA. Heavy DNA. It means 100% of heavy DNA are present. If we take out a ratio, like what is the percentage of heavy DNA, then it will be 100%. Later we will write answers for this as ratio of light DNA is to heavy DNA is to hybrid DNA here. We will write this one later. So after 20 minutes, this DNA got separated, means each strand will give rise to new DNA strand. That is the major concept here. So I am writing the heavy DNA which is having N15 and N15 in black color marker, and the new DNA strands that are formed in with red color. The new DNA strands are not having N15 containing nitrogen, but they have N14 only. Means the heavy DNA with N15 has given to new DNA strands that have N14 type of nitrogens only. If you write here the percentage of heavy DNA, answer is 0. But the DNA with one heavy nitrogen containing DNA and other with N14 containing DNA, both of them together are called as hybrid DNA. This is not called as heavy DNA because we have N14 nitrogen containing DNA and also N15 nitrogen containing DNA. So both of them together are called as hybrid DNA. Here we also can use another term, that is intermediate DNA strands. Intermediate density containing DNA. Now what is percentage of hybrid DNA? Again the answer will be 100%. I am not using the term like heavy DNA here. I am using the term hybrid DNA. Here when both strands have N15, I am using the term heavy DNA. E. coli again takes 20 minutes of time for its binary fission, but altogether from initial the timing is 20 plus 20. So by the time it has undergone second replication, the timing is calculated as 40 minutes. Now I am separating these 4 strands again. Black color one are 2 in number and new DNA strands are again 2 in number. So this is En15, En15. So this one and this one. And this is En14 and this is En14 containing strands. Always the existing N15 gives rise to normal N14 DNA only, because here in the test tube the medium taken is N14 containing nitrogen, the medium is having N14. Okay. That point I will write here. Medium has nutrient medium. Medium means nutrient medium. Has N15. This type of nitrogen. But here nutrient medium has N14. Here also after 40 minutes also again nutrient medium in which E. coli is grown has N14, not N15. So we will see what is the percentage of hybrid DNA and what are the names of these purely N14 containing DNA strands. Already we have given a term for the DNA strand with one heavy nitrogen, one normal nitrogen or light one as hybrid DNA. So this is hybrid DNA. This is hybrid DNA. But what is the name given for this one? N14 containing DNA strand is called as light DNA. This one is also termed as light DNA strands. Now, what is the percentage? Here we are getting 2 number of a hybrid and 2 number of light DNA. It means that it constitutes of 50% and it constitutes of 50, half number of hybrid DNA, half number of light DNA strands are obtained in the third type, means after 40 minutes. Now we have to give a term. These are considered as the original DNA strands with N14. After 20 minutes, we got F1 generation. F1 generation here is considered with a term that is generation 1. If we write here generation 1, obviously this will be generation 2 or F2 generation. So in F2 generation, we have got 50% of hybrid and light DNA. In generation 1, we got purely 100% of hybrid DNA. In original DNA strands, we got 100% of heavy DNA. Later, we will write the ratios like this: light DNA, heavy DNA, hybrid DNA of each and every generation. Again, after 20 minutes. In a sense, here once 20 minutes, here next 20 minutes, here next 20 minutes. Altogether constitutes here 60 minutes of time. Means after 60 minutes of continuously dividing DNA replication, DNA by the process of binary fission. What is the, again, percentage and the ratios here? So, here we have written here 50% we got in generation 2 or F2 generation. Then obviously it will be generation 3 or F3 generation. Now we will write how many number of hybrid heavy DNA strands are there? 1 and 2. Again, light N14 containing DNA strands are 1, 2, 3, 4, 5, 6. I am going to represent those one. This is heavy DNA strands and light one. 1, 2, 3, 4, 5 and 6. All because DNA is showing semi-conservation, means each strand will give a rise to new DNA strand, but that is protecting at the same time simultaneously its own genetic material. So I am separating this one because after DNA replication, each strand is behaving during the replication time as a template. So here we are getting new DNA. DNA replication also called as autocatalysis. So we will give terms for this one. This is N15 containing, this is N15 containing. All the remaining strands are N14 containing DNA strands only. So if we write, what is the number and percentages? So hybrid DNA is 1, hybrid DNA 2 in number. All the remaining are light DNAs, light DNA strands. If we take out from 100% of all these, how many strands are there in total? 1, 2, 3, 4, 5, 6, 7, 8. If 8 number of strands are considered as 100%, then how many number of hybrid strands are there? 1 and 2. What is percentage of 2 strands in that? If we do cross multiplication, 2 into 100 divided by 8, what answer we are getting? 2 ones are 2 fours, 4 ones are 4 25s. Means hybrid DNA is of 25%. It means that remaining light DNA strands should be of the 75%. Here we can discuss some MCQs. After 20 minutes, what is the percentage of hybrid DNA? 100%. After 40 minutes, what is the percentage of again hybrid DNA? 50%. After 60 minutes, what is percentage of hybrid DNA 25%, of light DNA 75%. Again, in generation 1 of semi-conservation method of DNA replication, what is percentage of hybrid DNA? 100%. If a question was asked, what is the percentage of light DNA in generation 1? Answer will be 0%, because here we did not see completely light DNA strands. After 60 minutes, if a question was asked, what is the percentage of heavy DNA? Or in generation 3, what is the percentage of heavy DNA? Answer is 0. Because here there is no purely heavy DNA. This is hybrid, which is having N15, N14. N14, N14 containing DNA strands are called light DNA. So there is no point of existence of heavy DNA after 20 minutes of time. Next we will see what is the percentage of hybrid and light DNAs after 80 minutes of time. After again 20 minutes of DNA replication by E. coli, that constitutes of 80 minutes of time. Here, how many strands all over we got? 2 number of heavy DNA strands, and 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So I am writing here 2 heavy DNA strands, but all the remaining are 1, 2, 3, 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And what about these two? 13 and 14. We know that this again gives rise to normal DNA. This is N15 and this is N15. So it is giving rise to normal DNA strands. That is light DNA strands. If we go out, what is name for the generation? This is generation 4. This is generation 1, 2 and generation 3, or F3 generation. So after 20 minutes we have got generation 1. After 40 minutes we have got generation 2. After 60 minutes it is generation 3. Correct only. So now it is generation 4 or EF4 generation. So what is the percentage of hybrid and light DNAs here, and how many number of double-stranded DNAs we got after 80 minutes? All over it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 number of total DNA strands are considered as 100%. Now, what about these only hybrid DNAs? This is hybrid DNA and this is hybrid DNA. So 2 number of hybrid DNA constitutes of what percentage? Again we do the cross multiplication. 2 ones are 2 8s, 4 2s are 4 25s. So 25 by 2, that is equal to 12.5% of hybrid DNA. So we will write answer here. Hybrid DNA is of 12.5% after 80 minutes. All the remaining constitutes, that is light DNA strands, are of 87.5%. The remaining one out of 100%. In this way, we can do the same after 100 minutes. What is the percentage of this hybrid and light DNA strands also? We can do same type of calculation. Now we will write here itself. The ratios. Up to 80% we can write. And you can calculate after 100 minutes also. In the same way how I have calculated of 60, 80, 40 minutes. In the same way you can calculate for 100 after 100 minutes also. So original DNA strands has 100% of heavy DNA. Now the ratio of light is to hybrid is to heavy DNA. Here we have got 0 is to heavy DNA is only 1. Now again about intermediate density DNA or intermediate density containing DNA. It is also called as hybrid DNA. Hybrid DNA. Now, what is percentage here? It is 100% of a hybrid DNA. Again, if you write the ratios, light chains and hybrid DNA chains and heavy type of DNA chains or strands. What is the percentage? In case of after, this is originally means at 0 minutes. This is at 20 minutes of time. Means after 20 minutes. So what is percentage after 20 minutes? When we are getting 100% of hybrid DNA, here we get light strands 0, hybrid strands again 100%, heavy strands again 0. Now, if we write the third one, this is F1 generation, we can write F1 or generation 1. If you write F2 or generation 2, that is after 40 minutes. How many number of DNA strands we are getting? Now directly we will write the percentages. Light is to hybrid is to heavy DNA is to heavy DNA. We have got here how many number of after 40 minutes? We got here light chains or light DNA strands 2 in number, hybrid DNA strands 2 in number, and heavy DNA strands 0 in number. This one can also be written as the ratio, cancel 1 and 1, 1 is to 1 is to 0. First of all, we have to check whether the ratio is asked in light hybrid heavy sequence or hybrid light heavy sequence or heavy light hybrid DNA strand sequence. We have to check what is the question asked about, what is the sequence. If it is asked as a hybrid is to heavy is to light, then it will be 1 is to 0 is to 1. But here I have written as light is to hybrid is to heavy. So I have written here answer as 1 is to 1 is to 0. Like this, we can calculate and write the number of ratios also. Here we have after 60 minutes, that is of generation 3, that is after 60 minutes. If we write the ratios of light is to heavy, hybrid is to heavy, how many light chains are there? 1, 2, 3, 4, 5, 6 are there. How many number of hybrid DNAs are there? 2 are there. How many number of heavy DNA strands are there? 0. So answer again we can write 2 1s, 2 3s. So it can also be written as 3 is to 1 is to 0. That is after 60 minutes in generation 3. Next, in generation 4, that is after 80 minutes of time. So here itself we have the proof. Number of, you write light is to, hybrid is to, heavy DNA strands. So heavy DNA strands are hybrid DNA strands are 1 and 2. So hybrid are 2, heavy is 0. What are the number of light DNA strands? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. If we do the cancellation, 2 1s are 2 7s are. So 7 is to 1 is to 0. Like this, the ratios varies from generation to generation and even after timing, basing on the timing. After 20 minutes, the ratio differs. It is 100% of hybrid DNA. No light, no heavy. After 40 minutes, 2 number of light strands, 2 number of hybrid DNA strands. 1 is to 1 is to 0. But while answering, if any such question has come, please check that what is the sequence or order of these light hybrid heavy DNA strands asked in the question. This is about semi-conservation of DNA replication.